again and go through the next presentation, which is on single cell dissection of schizophrenia reveals neurodevelopmental, synaptic axis, and transcriptional resilience. Again, both of them are bioRxiv papers and medRxiv papers, so these are all preprints. Your feedback is very welcome, and we'd love to keep improving the work. So Brad is the key person leading the study, and Shaheen and Jose have helped him with the computational analysis, and Sivan with the experimental work. So very briefly, this is a cohort of schizophrenia, bipolar disorder, and controls across a matched range of ages and across a matched range of BMIs. But the challenge, of course, with psychiatric disorders is that we actually treat them. So it's very difficult to get matching of the medications, because with bipolar disorder and schizophrenia, there are, of course, more medications. So, this is something that unfortunately will be a confounder for any such study, but this is something that we try to control for. The study, very briefly, is looking at the frontal cortex of 24 cases and 24 controls, and then dissociating these to create single nuclei tagged with droplets. And in this particular case, using a multiplexing approach that allows you to actually profile multiple samples simultaneously, which has the advantage of actually correcting for batch effects, because Brad has basically processed controls, schizophrenia, and bipolar jointly in every one of those experiments, these multiplexed experiments. We then interpret the data computationally and then analyze the differentially expressed genes, the drivers of GWAS loci, or at least predicted drivers, and the transregulators that likely, that are predicted to act downstream of that, and looking at what are the transcriptional factors that are associated with some of those genetic variants that control these enhancers and so on and so forth. So the obligatory plot for every one of these single cell studies is how are individual cells, shown as dots, falling with respect to each other in this giant graph projecting into a lower dimensional space. Many people use t-SNE and other tools for that. But in this particular case, we're not showing a two-dimensional space, we're showing a network of cells that are all connected to each other. So the underlying tool for this is ACTIONet. It's something that Shaheen Mohammadi developed in our lab, and in collaboration with several people, including Jose Davila and Sebastian Pineda. And that allows you to now look at this continuous distribution of moving from cell to cell and how they relate to each other in this network. We can then partition this network into closely connected subgraphs, and these are effectively the cell types and the cell states that are resulting from this. And you can see here a large diversity of excitatory neurons, of inhibitory neurons, each with different sub-annotations. So the excitatory neurons fall mostly by layer, and the inhibitory neurons by multiple different functional subtypes. You can see also a lot of the macroglia, such as oligodendrocytes, astrocytes, and OPCs, and also the microglia. And then you can see how these relate with different previous annotations, and how the different markers of these cell types are enriching in different parts of the space. What I really want to point out is two cellular states that are captured in addition to the distinct cell types. The difference with the cell states is that they are not quite a cell type. They're kind of cross-cutting sometimes across multiple cell types. So there's one cell state of the neurogranin-enriched neurons, and then another cell type state of these SZTR neurons that I'm going to talk about shortly. And you can see how these are cross-cutting across multiple of these cell types. What's really interesting about neurogranin is that it actually is depleted for schizophrenia individuals. And what's really interesting about ACTR is that it is enriched in schizophrenia individuals. So this is kind of interesting. And if you look at what are the composition of archetypes, you can see that they're very different between different donors, with some of those cell states being greatly altered. But now we can start asking, where are the differentially expressed genes occurring? And what you can see.
I got a question.
Yeah.
Which is, from the best that we understand from neuroscience, is that a reasonable finding that the cell types are stable, but it's rather their activity that's going to differ?
Yes, indeed. We don't find dramatic changes in cell type composition between individuals. If you look at cases and controls, they're roughly at 50-50 between most of these cell types, the only exception being SZTR and neurodevelopment.
Got it. Thank you.
So now we can start asking about differential gene expression in schizophrenia. And then what we can see is that there's a lot of genes that are overexpressed in this SZTR cell state. And there's a lot of genes that are downregulated in layer 5 and layer 6 excitatory neurons. And this continues this way. So basically you can see that a lot of the downregulation happens in these excitatory and inhibitory neurons. And there's very little change that is happening actually in the glial cells. More or less the way that we would have expected, but now we can see that single cell resolution. Basically, people previously looked at global changes based on global changes based on the convolution of bulk data. This is the first time that we can actually see that at single cell resolution. And now we can basically ask, what are the differentially expressed genes? You can basically see the differential expression both for overexpression and for under expression in schizophrenia across all of these different genes, and also the cell types in which they act. And you can see, again, this enrichment for excitatory and inhibitory neurons, but much lower for the glial cells.
So my question, this is restricted to those genes that are statistically differentially expressed? Is that what it is?
Yeah, using the same threshold of differential expression, we can look at how many differentially expressed genes are there, how many are upregulated, downregulated, and so on and so forth. And then,
but I'm saying in that particular figure, how do you select those particular genes?
Oh, Brad, do you want to answer that question?
Yeah. These are not the full extent of the differentially expressed genes. So the differentially expressed genes were called pretty liberally because we're more interested in kind of gene sets and associations at that level.
Yep.
So all of these 8,400 differential expression events across the different 20 cell populations are raw p-value less than 0.05, fold change greater than 0.1, log fold change. And this was a selection of genes that were both the most consistently dysregulated across populations, and then the genes that showed specific patterns where they're differentially disturbed, upregulated in excitatory, downregulated in inhibitory, genes that showed a particular pattern that came up quite often where they're overexpressed in the SZTR cell state while being downregulated across other neuronal populations. So the selection of these genes to display here is not a robust single criteria.
Got it, thank you.
Okay, so that's at the level of individual genes, and then we're going to talk a lot about pathways in the next few slides. So the first thing we can ask is, what is the consistency of these transcriptional changes across different states? Namely, if you look at the genes that are differentially expressed in layer 2 and 3, are they the same as the genes that are differentially expressed in layer 5 and 6? So the off diagonals here show the comparison between every pair of cell types, and then you can see here how the ranking in fact corresponds. The closer you are to the diagonal, the more agreement there is. And then you can see that there's in general, good agreement between excitatory neurons, between inhibitory neurons, and also across neurons, but much less with the glial cells. We can then do something very cool. We can basically take, and pay attention to this because this is a complicated part. We can take the differential expression that we observe for every one of those cell types between schizophrenia and control individuals. So for layers 2, 3, we see a global shift in a bunch of genes. We record it. And for that one, we record it and so on and so forth. So basically, there's the set of differentially expressed genes in every one of those columns. And then we can go back to every individual and ask, how is the expression difference of that individual varying compared to the average of all the other individuals? So for every person, for example, for SZ7, we ask, how is SZ7 deviating from the average of all of our individuals? Is everybody with me so far? So the first component is, how is the cell type shifting across all genes? And the second one is, how is that person shifting across all genes? And then we look at the correlation of the two. If SZ7 differs from the rest of the people in the same way that this cell type differs between cases and controls, then we paint it red. If it varies exactly the opposite way, we paint it blue. So that allows you to predict for every person how is their global transcriptional profile related with schizophrenia cases or controls. With me? Perfect. So that allows us to now start sorting individuals according to their transcriptional pathology ranking. So this is basically asking at the transcriptional level how schizophrenia-like or how control-like is every person. And what's really cool is that it actually allows you to partition individuals according to cases, controls, and somewhere in the middle. And you can see this red gradient going down because we've ranked individuals by that gradient. Here's what's really spooky. If you look at some schizophrenia cases, they actually have transcriptional profiles that are very normal looking. But those are the ones that are, in this column here, associated with this SZTR cell type. So the individuals that have the most abundant SZTR cell type somehow are indeed cases, but their transcriptional profiles are control-like. There's many ways to interpret this, but one way that we present in the paper is that perhaps this SZTR cell state is a transcriptional resilience cell state, hence the name, where if you are an individual who has that cell state, then the other cells might actually be more likely to be transcriptionally normal. And we welcome other interpretations, but this is one interpretation that we put forward in the paper. And indeed, if you look at the transcriptional pathology score, schizophrenia patients are high, control patients are low, but for some reason, these SZTR people are even lower than low. And we are as intrigued as you are. And I wish we had a better answer, but this is science. We say what we find. We say one interpretation. We might be wrong in the future, but at least people are paying attention to it.
Manolis, do you have access to the clinical histories of these patients?
Fantastic question. That's exactly where we went, Zach. So we basically went through the histories of these patients to try to see what are the indications. Are the symptoms perhaps less severe, and so on and so forth. We don't have an answer yet. And in fact, Brad, I don't know if you've had a chance to actually go through in detail for all of them, but at least we looked at all of the quantified variables. We couldn't find any differences, but I'd love to sort of hear from Brad on the actual clinical notes.
So I have looked through the records, and so I work at the Brain Bank. It's part of what I do here at McLean. I'm the associate director, and this is a complicated issue as far as the consistency and the completeness of postmortem records and what we are able to glean from them. And so I have looked through the records, and this is something that I'm still thinking of, but what I've on my work so far, there are no indications that are consistent across these cases that they have either a more severe or a more light experience of the symptomatology of disease.
Were any of these patients partners, healthcare system patients?
So these are brain donors, people who donated their brain to the Harvard Brain Tissue Resource Center here at McLean Hospital, but they are from anywhere across the US. I
see. Okay, and another possibility, of course, would be which medications they were on.
We also looked at that, and we did not find a difference in medications either. Basically, what we were wondering is, could SZTR be indicative of a cell state that is a result of medication, which is also successfully bringing the transcriptional profiles down? We did not see that either.
And that's one thing that I'm can be more confident in our ability to extract from the records, a quantifiable metric like medication. And yes, there's no clean association with exposure to any amount or specific class of medication.
You might want to also stratify your analysis and repose the same questions, but look separately at males versus females, because certainly in other diseases like autism, the males are much more homogeneous than the females, and for a variety of interesting reasons. And so you might find a clear signal in one or the other. And sorry for the squeezy sound. That's my dog squeezing a squeeze toy.
So Ariel is asking a question about the significance of these numbers of dots. We are now, we have a meeting tomorrow with Panos Roussos, who's also done single cell profiling in schizophrenia, to basically see if we can detect this SZTR cell state in his data as well. And we will report back as we increase the count. So, we are part of the Second Code Consortium. So, there's only one other group who's looking at that. And then we are, connecting with them. Peter Kurchenko is also part of another single cell study that was posted also in medRxiv, I think, a few weeks after ours. And we also plan to somehow compare notes and see if the data replicates across. All I can say is that it's intriguing, and we're, sort of trying to replicate it and see where else it holds. I want to point out one other exception. So you see this control individual who's all the way up. So this control patient, even though was not diagnosed as schizophrenic, has very much schizophrenia-like expression profile. And indeed, his son is schizophrenic. So, we might have escaped it in your lifetime, but maybe the, genetic predisposition is reflected in a sort of above threshold diagnosis for the son. So, again.
I mean, that's a useful speculation, and it'll be hard to tease it apart with the data they have. But you could imagine, for example, that that most of these people all have this disorder, but some are more dependent on a stronger environmental push because the heritability is, I think, pretty high, but it's not 100%.
Exactly.
And so there might be certain individuals who were not at great risk, but they got pushed there by environmental conditions or psychodynamic conditions.
Completely agree. So yeah, that's why we basically see this gradient. And when we look at polygenic risk scores, for example, they're not perfectly recapitulating this. The polygenic risk scores are indeed enriched in the schizophrenics, but not it's not a slam dunk. Genetics is not everything. Environment plays a huge component. All right, so then we went off and did some validations using RNAScope to sort of show that indeed there's specific selected genes that are higher in schizophrenia or higher in controls or higher in SZTR. And indeed, this all validates very nicely, and you can see this gradation across them. The last couple of points that I want to make is that if you look at the gene ontology enrichment for the upregulated and the downregulated differentially expressed genes, you see this enrichment in synaptic signaling gene ontology terms, in synaptic organization, and neurodevelopment. So this is directly looking at the differentially expressed genes. Then you can also go into the GWAS for schizophrenia, and you basically have these 100+ loci across the genome that are now robustly associated. And what we can do is look within each of those loci for, are there high C, basically linking evidence between the GWAS locus and the differentially expressed gene for some of them, or simply proximity for others, the ones in white, that allows us to predict what might be the differentially expressed gene in the locus and the direction in which it changes. And we've annotated those, the positively associated with schizophrenia in red and the negatively with blue, and also the cell type in which it changes according to the corresponding color of that cell type. We've put a circle in the cell type of highest differential expression, and also colored according to plus or minus every single cell type where you see this change. And you see this very strong association with SZTR, as I mentioned earlier, the strong association with excitatory neurons, weaker with inhibitory, and very low for glial, enabling us to now start gaining some candidate target genes and the directionality and the cell type where they might be acting for each of those GWAS loci, enabling us to start thinking about sort of therapeutic avenues targeting those genes. We can also go beyond schizophrenia and ask what is the enrichment between genome-wide association study variants associated with schizophrenia, bipolar, mood disorders, ADHD, and autism spectrum disorder, as well as Alzheimer's as a control, and basically see, are they enriched with the cell type specific dysregulation that we observe in schizophrenia? And indeed, we find that the strongest GWAS enrichment is for schizophrenia variants, but also we see an enrichment for several of the other psychiatric disorders, but not for Alzheimer's, which is sort of reassuring control, and not for any of the glial cell types, which is again a reassuring control. So that basically says that indeed there is shared disruption between the schizophrenia changes and the genetic variants associated with those, as one would expect, given the common risk patterns between these different disorders. Now we can go upstream to what are the regulators and basically start looking at what are the transcription factors that target these differentially expressed genes that are enriched in neurodevelopment processes. And indeed, we see that even though the differentially expressed genes fall in all three classes, the regulators of differentially expressed genes focus much more on neurodevelopment and, to some extent, synaptic organization. So this is kind of cool. So it basically says that indeed there's a developmental component that might happen at the master regulator level, even though the differential expression happens, at multiple genes nearby.
Manolis, just speaking for myself, I lost a thread. How do you determine whether these things are regulators?
No, you didn't lose the thread. I forgot to say it. So, yeah. Brad, could you remind me? So we now have the differentially expressed genes. The regulators, are they based on motifs? Are they based on ChIP-seq? I don't remember what they're based on.
So this is, associations are made using the CHIA3 tool, which looks at overlap of gene sets. So that differentially expressed genes within each cell type, do they enrich gene sets known to be from a prior experimental work associated or targeted by these transcription factors?
Thank you, Brad.
I thought you were going to do the grand unification theory with the prior paper and looking at.
That would have been lovely, but we didn't do that.
Yeah.
Yes, we actually didn't have the results when we were doing this study. Now that we have both, we should probably repeat it with that.
Yeah.
Okay, so what we're finding is that a subset of these master regulators of these differentially expressed genes are themselves GWAS-linked genes. So the regulators are themselves GWAS hits. And that's sort of alluding to that very first slide that I showed you, where the transcription factor is in a GWAS locus, and it is in fact targeting many of these genes that are differentially expressed, which is more or less what you would expect. Basically, some of the strongest genetic effects are coming from master regulators that are having a trans effect rather than just cis-only effects. And we can now take those regulators and group them into modules using the correlation of expression of the transcription factors across different neuronal cell types and across all individuals. And what you can see is, there's like 1, 2, 3, 4, 5 modules that are including multiple of these regulators. And then we've named these modules according to the master regulators that are, sort of most appealing within these modules, even though you can see this global correlation with multiple factors. Our favorite module so far is also the largest, which is this MEF2C SADB2 module, which in fact contains multiple GWAS hits for schizophrenia and multiple GWAS hits for NDD, and multiple that are in fact both. And you can see this module here, this MEF2C module, also overlaps very nicely with this ME37 module that was previously discovered by SZTR. And if you look at the functional enrichments for the different subsets, the intersection of the two is enriched in neurodevelopment, but also synaptic plasticity. And then, the outsides are slightly differently enriched. So we then went and actually used cut and tag to map the transcription factor binding sites using postmortem human neurons, and looked at where are the targets of these happening. And indeed, we do see this enrichment for the targets of the experimentally defined ChIP-seq, like targets using cut and tag are indeed enriched in synaptic and developmental processes, confirming this sort of synaptic neurodevelopmental axis that we mentioned in the title. And then the transcription factors that are enriched in these differentially expressed genes are also very noteworthy for being active both in the developmental and in the adult stages, where you can basically see, is this factor, are the TF-bound genes enriched with up or down regulated genes in each of these cell types and each of these regulators? And also, where are the regulatory elements active using psych-encode data of different stages of development? And what we can see is that the targets of these SZTR cells are in fact active in both, suggesting perhaps this connection between fetal and adult. So just to summarize, we basically looked at cell type specific DEGs that are agreeing with prior work, but also a lot of differential expression in specific cell types. We find that these differentially expressed genes are predominantly impacting these deep layer cortical projection neurons and these PV expressing interneurons. We find that 68 of the 145 GWAS loci are in fact linked to DE genes in at least one cell type, suggesting candidate genes, the direction of effect, and the cell type of effect. The differentially expressed genes implicate both postsynaptic density, synaptic plasticity, and neurodevelopmental pathways, but the transcription factors that target those enrich specifically neurodevelopment. The SZTR cell state is anti-correlated with these transcriptional signatures of schizophrenia in the other cell types, and they're enriched in schizophrenia and control individuals with the least SC-like transcriptional states. And then the upregulated schizophrenia differentially expressed genes are associated with fetal enhancers, and downregulated DEGs associated with adult or shared enhancers, as we see here. So the upregulated genes enrich in fetal enhancers, and the downregulated genes enrich in adult enhancers, except for these common one. So as I mentioned earlier, this work is led by Brad in collaboration with several other both computational and experimental folks. And we really thank all of the folks who have donated their brains to make this study possible. And this is the support for the grant. Okay.
So Manolis, that's a really, that was a wonderful tour de force. Unfortunately, we're past the hour. And because in the era of Zoom, I'm very respectful for Zoom torture. I'm going to preemptorily bring our meeting to a close, even though there may be more questions, just because I know people have to go onto their next Zoom. So Manolis, thank you. That was really, really helpful. And frankly, you gave me personally a lot of food for thought and ways that we might be able to collaborate in the near future. So thank you.
Beautiful. Thank you so much for having us, Zach. Again, this is an extremely collaborative team. We're super thrilled to collaborate. We work on a huge range of problems, and we're eager to collaborate with basically, I think, Zach, what you have in your department is really very unique, and it's a group that we're super, super excited to interact with. So I'm very, very happy to see so many people here and eager to find ways that we can interact and work together. Because frankly, we ask a lot of the same questions from very complementary approaches, and that's the secret ingredient to good collaboration. So, diverse takes on common passion.
Exactly. Well said.
Awesome. Thank you.
Thank you.
Thank you all. And please email us. We're super friendly and excited to interact. Bye-bye.
Thank you. Bye.
Brad, we should chat soon. I have another meeting coming up, but I could be free at 6. So
I just sent you a text to your text earlier in the day. So I will